So in 4.12, this is kind of a long problem, I think. So we're working with 3 gene pairs. Let's take a look here. The first pair exhibits incomplete dominance with a flower color. So flowers can be red. Oh, I want yellow here. Sorry. Red, pink, or white. Okay, and let's use, what can we use for this? Let's use little w. So incomplete dominance is going to look something like this. We have 2 big W alleles, be red, a big W and a little w, you're pink, and you got 2 little w's, you're white. So the second gene pair, the second pair, leads to personate, and I guess it's the flower shape. Personate is the dominant trait, and peloric is the recessive trait. Again, I'm not really sure exactly what those traits are, but we'll use little p's for peloric and at least 1 big P to be personate. And the third pair produces either tall or dwarf. Okay, this is easy. So tall, at least 1 big D allele, and dwarf, 2 little d's. So now for the question here, homozygous plants that are red, personate, and tall are crossed to those that are white, peloric, and dwarf. Determine the F1 genotypes and phenotypes. Okay, so red, 2 big W's, personate, 2 big P's, peloric, 2 little p, tall, 2 big D's. And white, 2 little W's, peloric, 2 little p's, and dwarf, 2 little d's. Now these can only make one type of gamete, right? Big W, big P, big D, and these can only make 1 little W, 1 little P, 1 little D. So when we cross these together, all the plants are going to have a big W, big P, big D, little d genotype. Now what's the phenotype here? Well, big D is dominant to little d, so they're all tall. Big P is dominant to little p, so they're all personate. And big W is dominant to little w? No. The question says it's incompletely dominant. We can go up to our little chart and we can see big W, little w, genotypes are pink. So we have pink, personate, and tall. Okay, so that's the answer to the first part of the question. Now the second part of the question is if the F1 plants are interbred, what proportion of the offspring will exhibit the same phenotype as the F1 plants? Okay, so we want to know if we cross these plants together here, these genotypes, how many of the offspring are going to have this phenotype, tall, personate, and pink? What proportion? 1 out of 16, or 1 out of 32, 5 out of 10, what's 1/2? We have to figure that out. And so I think the easiest thing to do here is, start with the forked line method, right? So split these into individual pairs of genes and start going through the genotypic ratios and combining them. So let's just look at, pretend we're working with a cross with only W's, with only the gene for flower color. So if we cross a big W, little w plant to a big W, little w plant, we would know of all the offspring, 1/4 would have 2 big W's, 2/4, sorry, I'll neaten that up a bit. 2/4 would have a big W and a little w, and 1/4 would have 2 little w's. Okay, so now we could look at the other, one of the other gene pairs here. So the one for personate or pyloric. Again, we know of all the plants that have 2 big W's, 1/4 are going to have 2 big P's, 2/4 are going to have a big P and a little p, and 1/4 are going to have 2 little p's. And how do we know that? Because we've memorized Mendel's monohybrid ratios in the F2 generation, or when you cross heterozygous plants that are heterozygous for a pair of genes, for a pair of alleles here, big P and little p, and we know this ratio, so we can just plug that in, or we could do the Punnett square and we could figure out what the ratio has to be. And we could do that again for big D and little d up here. So of all the progeny that inherit 2 big W alleles, 1/4 of them are going to inherit 2 big P alleles, and 1/4 of those are going to inherit a big D, 2 big D alleles. 2/4 are going to get a big D and a little d. 1/4 are going to get 2 little d's. So this problem could get really long, but again, we have a specific goal here, right? We don't need to know the ratios of all the different genotypes that are possible from crossing these 2 plants, right? We don't need to know the genotypes of all the progeny from this cross. We only need to know the genotypes of all the progeny that are going, or how many of the progeny are going to be tall, personate, and pink. So any offspring that inherits 2 big W's is going to be red. So we don't need to worry about any of those. And for these down here, any offspring that inherits 2 little w's, they're all going to be white. So you don't need to worry about those. We only need to worry about those offspring that have a big W and a little w. So let's take a look at those. Of all the offspring that inherit a big W and a little w, now 1/4 are going to inherit 2 big P's. 2/4 are going to get a big P and a little p, and 1/4 are going to get 2 little p's. And let's see here. Now we want personate and pink. So again, of the 2/4 progeny that from that cross that inherit a big W and a little w, we don't care about the 1/4 of those that are going to be pyloric. So these would be pyloric. We don't care about those. So we don't need to continue on with that genotype down here. We only want the tall and personate. Okay, so let's take a look here. So these 1/4, 2 big P's, 2/4, big P, little p, those are the personates. So of the offspring that inherit a big W and a little w and 2 big P's, 1/4 are going to inherit 2 big D's, 2/4 are going to get a big D and a little d, and 1/4 are going to get 2 little d's. Now these are dwarf plants, right? And we don't even care about those. So we're going to drop that out. We don't care about those. We do care about these 2/4 that are personate though. So 1/4 of those are going to be 2 big D's, 2/4 are going to inherit a big D and a little d, and 1/4 2 little d's, and we don't care about those, so we'll cross that out. So after all that, what do we care about? Let's see. We want, okay, 2 quarters are going to have big W, little w, 1 quarter, big P, 1 quarter, big D, big D. Okay, so let's multiply those across. We should have 2 out of 4 times 4 is 16 times 4 is 64. 2 out of 64 are going to be big W, little w, big P, big D, big D. And then for the next one, 2 out of 64 are going to have big W, little w, big P, big P, big D, little d. And for the next one, multiply this to there, 4, right? 2 times 2 is 4, so 4 out of 64 going to have big W, big P, little p, big D, and I can see I messed up right here, so this should be 4 out of 64 right there. And then for the last one, we're multiplying down to there. So 8 out of 64 are going to be big W, big P, big D, little d. So we should have 6, 10, 18 out of 64, or 9 out of 32 should have the same phenotype. Is that right? Let me see. 1, 2, 3, 4, 5, 6. Let me see. 2, 6, 10, 18. Okay. 18 out of 64. So 9 out of 32 should have the same phenotype as the F1 parents. So, okay, so that was kind of a long problem, but we were able to take a shortcut just by focusing in on those particular genotypes we needed to see in the progeny of that cross.